Welcome to this week's episode of Brainstorm, where we give you a glimpse into the world of science for Wednesday, October 2nd, 2013. Our top story comes from the world of biotechnology. Some scientists from Harvard University have discovered an additional layer of complexity within genetics that may allow for enhanced engineering. As we have mentioned on the show before, when it comes to the synthesis of proteins, not all codons are created equal. As a refresher, codons are set to 3 nucleotides in messenger RNA that correspond to an amino acid while a ribosome is synthesizing proteins. There are way more codons than there are amino acids, so many amino acids have what are called synonymous codons. However, even though synonymous codons mean the same thing as far as the ribosome is concerned, some are much more common in the genetic code than others. Synonymous codons that are not as abundant in the genetic code are referred to as rare codons, and previous work showed that they have unique properties. If a gene has rare codons toward the beginning of its sequence, organisms seem to produce more of the protein for that sequence. There were 2 hypotheses as to why this was the case. The codon ramp hypothesis suggests that the rare codons actually slowed the initial synthesis of a protein, which ends up making the process smoother in the long term because synthesis that starts out fast will jam it all up. The other hypothesis said that the rare codons tended to cause the RNA strand to fold in a way more conducive to translation, but did not intrinsically increase speed. While the codon ramp hypothesis seemed true, seeing as an initial experiment showed that rare codons did in fact increase protein production by up to 60 times, a follow-up experiment disproved the codon ramp hypothesis. They showed that even if the sequence had rare codons but folded improperly, the mRNA resulted in less protein production. An mRNA that was designed to fold well but had common codons still resulted in more production, meaning that the way mRNA folds is crucial to protein production and not which synonymous codons are used. This gives all the scientists a new tool when designing genes for medicine and industry, greatly increasing the potential of producing renewable products through biotechnology. Next is an update from the world of biology as it applies to ecology. Have you seen the episode of The Simpsons where Bart introduces an invasive species of lizard and then they get a species of snake to kill it? Well, some researchers from the University of Rhode Island are basically doing that, but with a plant and a moth. All joking aside, the scientists from Rhode Island have been studying a particular species of moth from the Ukraine for use as a biological agent to control swallow-wort. They raised a population of the species in the University of Rhode Island's insect quarantine laboratory, and the caterpillars of the species seem to exclusively eat invasive weed. This is encouraging because it can grow in a number of environments and has spread to many locations throughout the northwest United States and up into Canada. Monarch butterflies often lay their eggs on it, but the plant is toxic to their caterpillars. The release of the moth is likely to be approved by the USDA for next spring, but Canada has already approved the moth, so the scientists have sent some specimens to colleagues there. And results are already promising for the control of swallow-wort in the Ottawa area. Hopefully this will help control the weed as the moth does in Europe and doesn't end up going horribly, horribly wrong. Well, hope you enjoyed this episode. What creative methods would you use to control an invasive plant or animal species? Let us know your thoughts on that and all the stories in the comments.